Good afternoon, dear students. Now let us continue with the recombinant DNA technology. Till now we have studied what are the different types of restriction enzymes. Okay, what are restriction enzymes? What are the different types of DNA manipulating enzymes? And why they are required, DNA manipulating enzymes are required for recombinant DNA technology experiments. Then what is gene cloning? What are the different types of vectors that are used for cloning of a foreign gene into a cloning organism? Now let us today discuss how a recombinant DNA. What is a recombinant DNA? Here we are going to discuss about recombinant vectors. What is a vector? It is a carrier DNA molecule like a plasmid or a bacteriophage or a cosmid. Now what is a recombinant vector? The vector having the foreign gene to be cloned inserted inside it. So now today we are going to discuss how to introduce the recombinant DNA, rDNA means recombinant DNA, into a host cell, that means into the cloning organism. So what are the different methods of introducing recombinant DNA into host or cloning organisms? So after the recombinant molecule has been generated, that means after you have selected the foreign gene to be cloned and you have inserted the foreign gene inside the vector with the help of restriction enzyme and DNA ligase, what you have is a recombinant vector. So the next step is introduction of the recombinant vector or a recombinant DNA into a cloning organisms. So, and the type of, you can say the method that is used for introduction depends upon various factors like the type of vector, the type of host cells and so on. So, what are the different methods that are used for introduction of recombinant DNA into a host organism? The first and the most widely used method for introducing recombinant DNA into the cloning organism is transformation. In recombinant DNA technology, the most commonly used method to introduce recombinant DNA into a living host is called as is transformation. The most commonly used method is transformation. What is transformation? Unilateral, unidirectional uptake of foreign DNA fragment by the host cell. Okay, so unilateral uptake, incorporation and expression of a foreign DNA molecule, right, takes place in transformation. In the process of transformation, the bacterial host, or you can say the cloning organism, takes up the naked fragment of DNA from the surrounding environment naturally. Okay, so here the cell which is going to clone the foreign gene, that is the bacterial host, takes up the DNA from the surrounding environment naturally. But not always. But a bacterial cell will not always take up the foreign DNA or take up the naked DNA inside it naturally. Sometimes the organisms are not competent to take up the DNA from the environment. In that cases, host cells like E. coli, yeast, mammalian cells do not readily take up the foreign DNA. Why? Because they are not competent. And they have to be treated chemically to become competent so that they will accept readily the foreign DNA fragment from the surrounding environment. They will take up the recombinant DNA from the surrounding. Okay. So transformation is unilateral uptake of DNA from the surrounding, incorporation into the genome and expression of the foreign gene. Okay. So a bacterial host takes up the DNA from the surrounding, the recombinant DNA from the surrounding naturally, but when the cell is not competent, it will not take up the foreign DNA. And hence, in that case, the cells have to be treated chemically. Which chemical? Especially calcium chloride treatment is given to, say, like cells like E. coli, so that they become competent, they become permeable, and they take up the recombinant DNA. In 1979, okay, it's written 70, but it's almost 79, Mendel and Higa found that E. coli cells become markedly competent and take up the external DNA when they are suspended briefly in cold calcium chloride solution. That means CaCl2 treatment is given so that the cells become competent, they become permeable, they will readily accept the foreign DNA, that means the recombinant DNA. The second procedure for introduction of recombinant DNA into host is transfection. See, transfection is almost similar to transformation but carried out in eukaryotic cells especially. Okay, here also the foreign DNA is taken up, okay, and it's taken up and the foreign DNA is expressed, its products are produced and all. But when the recombinant DNA is introduced into bacterial cell, is a phage DNA rather than the bacterial or plasmid, the process is called as transfection. Okay, so when the recombinant DNA introduced into a bacterial cell is a phage DNA rather than a bacterial DNA or a plasmid, the process is called as transfection. Here, the foreign DNA may be coated with various chemicals like calcium phosphate or so. Thus, the process of transformation of DNA by a phage particle is called as transfection, and that too especially in eukaryotes, especially in plants. Number 3, the third method is transduction. It refers to transfer of bacterial DNA from one bacterial cell to another by means of transducing phages. We have studied genetic recombination, transformation, transduction, conjugation. So what is transduction? It is a process in which we transfer a bacterial DNA from one bacteria to another with the help of transducing phages, that is transduction. Now here diagrammatically we have shown how a recombinant DNA is put into a host cell. Say for example, we have here a recombinant plasmid. What is a recombinant plasmid? This is a plasmid having foreign gene inserted inside it, which is shown in red color. Okay, so a foreign gene is inserted inside the plasmid, so now it's a recombinant plasmid. The recombinant plasmids are mixed with E. coli host. If the E. coli cell is not competent, it will not accept the plasmid DNA. So we have to treat the E. coli cells. These E. coli cells, if they are not competent, they are treated with calcium chloride. They are treated with calcium chloride. Okay, CaCl2. So when they are treated with calcium chloride, they become permeable and they can readily accept the foreign DNA. And the E. coli host, when it accepts the recombinant DNA, the resulting cell is called as a transformed cell. The cell is called as a transformed cell. Okay. The next method is microinjection. It is a type of physical method because in this method, what we do, we use a fine pipette. A fine pipette is used to insert recombinant DNA directly into the nucleus of the cells. Again, practiced mostly in eukaryotic organism. Okay. So, it is a physical method where we use a micro injection like a needle. Okay. So, we have use of, this is a fine pipette or a, you can say syringe, which is used to insert the recombinant DNA is directly inserted inside the nucleus of the host organism, nucleus of the cloning organism. This technique was initially applied to animal cells, okay, but now this is successfully used even in plants. So eukaryotes, especially animals, in case of animals, this microinjection technique was used where the recombinant DNA with the help of a needle syringe is directly, fine pipette is directly injected inside the nucleus of the host. Okay. Now this was basically used for animals, now widely used for plants also. The next is microprojectiles, also known as ballistics, or use of gene gun to insert. Using a gene gun to insert foreign DNA, that means recombinant DNA inside the host organism. The second physical method, again this is used mostly in plants. The second physical method involves bombardment of cells with high velocity microprojectiles. Okay, usually particles of gold or tungsten that have been coated with DNA, right? So what are microprojectiles? They are small molecules. It's a physical method again, small molecules. What are those? They are gold or tungsten molecules. They are coated with the recombinant DNA. Rather, we can say the recombinant DNA is coated with the gold or tungsten particles. They are small fine particles, and with the help of a gene gun, they are, or with the help of a particle gun, they are bombarded on the nucleus, or they are bombarded on the cells. These microprojectiles are fired at the cells. They are fired or bombarded on the cell with the help of a particle gun. The velocity or speed of bombardment is so high that it directly crosses the cell wall, cell membrane and gets inserted inside the host cell. These microprojectiles are fired at the cells from a particle gun. This unusual method is termed as ballistics and have been used in number of different types of cells, especially in plants. Now, this is a remarkable method which was developed to introduce foreign DNA into mainly plant cells by using a gene gun or a particle gun. Microscopic particles of gold or tungsten are coated with DNA of interest and they are bombarded onto the cells with a device like a particle gun, and hence we use the term ballistics, right? You can see a diagram here. There is a firing pin here, right? These are the microprojectiles, that means gold or tungsten particles coated with the DNA. This is a firing gun, okay, and these are the target cells. And these are the microprojectiles which are filled into the gene gun. Now, as soon as the pin is fired, the pin is pressed, the microprojectiles are fired on the target cells and they directly enter inside the target cell. So, sure-shot method of introducing the foreign DNA into the host cell. The next method is electroporation, in which electric current is given. So, by applying short electric pulse to the cells, small pores are created on the cell membrane. Okay. By applying short electric current, small pores are created on the cell membrane, and then through this cell membrane, the recombinant DNA can easily enter inside the cell. That is called as electroporation. Right. So, in this diagram, we have shown various methods. Like this one is transformation. Here we have written transformation, where the host cell is competent and it will take up the foreign DNA on its own, or if it is not competent, it is made competent by giving calcium chloride treatment so that it accepts the foreign DNA fragment. Okay. Another method is like this is virus. This is a virus particle. So, by the method of transduction. So, you can see this is a virus particle. So, with the help of the virus, the foreign DNA is inserted inside. That means by a process of transduction. Then we've studied micro-injection method. Okay. So, with the help of the needle, or you can say micropipette, the DNA is directly inserted inside, or with the help of this is a particle bombardment gun, and DNA coated tungsten particles are directly inserted inside the cell. So, these are the various methods by which a foreign DNA or a recombinant DNA can be introduced into the host organism. And what we have written is a transgene here. That means the foreign gene to be cloned is inserted inside the bacterial chromosome, or actually it is present inside the vector. So, it's a transgene. So, that is all about various methods used for introduction of recombinant DNA into host cell. In some of the plant cells, like when we are constructing Bt cotton, one more method can be used. Another method of introducing foreign gene is by a natural genetic engineer, that is by use of Agrobacterium tumefaciens, especially practiced in plants. Okay. So, these are the various methods of introducing recombinant DNA into host cells.